Hello and welcome to this podcast from the BBC World Service. Please let us know what you think and tell other people about us on social media. Podcasts from the BBC World Service are supported by advertising.
You know, in life, no matter how much you are down, if you are willing to give your best and fight, you can always have a fighting chance to come back.
We had to rise from the ashes.
Search for amazing sports stories wherever you get your BBC podcasts.
Welcome to Science in Action from the BBC World Service with me, Roland Pease. Later in the programme, it took him almost 50 years to get the Nobel Prize, but his name will be memorialised in physics as long as it's studied.
Professor Higgs, I now ask you to step forward to receive your Nobel Prize from the hands of His Majesty the King.
Peter Higgs, who's just died, will be having an appreciation a little later. At the grubby end of physics, incredible claims of high-temperature superconductivity have been called falsified and fabricated by a university inquiry. We'll be hearing from the journalist who's been digging away at this scandal. And the single-cell seaweed that's conquered the chemistry of capturing nitrogen straight from the air.
With this, these algal cells are now capable of both carbon and nitrogen fixation, so they can kind of make all the nutrients they need.
A unique evolutionary achievement. We start, though, where we keep going, the continued spread of highly pathogenic bird flu through species and around the world. It was at the end of a special edition exactly a year ago we heard warnings of the havoc the virus could do if it got into the densely packed iconic bird colonies on Antarctica. And then at the end of February, Spanish virologist Antonio Alcami gave us genetic confirmation that H5N1 had indeed arrived on the continent. And then Antonio got in touch just after to say he was taking his pop-up biology lab on board the research ship, the Australis, to see how far the virus has spread. He's still flying back, but the mission was led by Australian bird specialist Megan Dewar. And in short, Megan, how worrying was what you saw?
Unfortunately, we detected a number of new cases of avian influenza on our expedition through the Trinity Peninsula and down to the Weddell Sea, and unfortunately also detected a mass mortality event due to the virus.
A mass mortality event, just describe that. What do you mean?
So at a breeding site for skuas, so the colony roughly was around about 130 birds, 50 of those were deceased.
Were the survivors, what was their condition? Are they still vulnerable? Could you tell how recently it had struck?
A lot of the carcasses were extremely fresh and most likely died anywhere from a couple of days before we got there to within a couple of hours of us arriving. So it was an active outbreak. And although there were a lot of apparently healthy birds flying around, the rest of the colony was still definitely at risk of bird flu.
Which birds again was that?
Skuas, so both brown and South Polar skuas.
And they're scavengers, are they?
They're predatory scavenger birds, so they will prey on penguins and penguin chicks and eggs during the season, but they'll also scavenge on carcasses as well.
What about penguins? They're the iconic species.
Yes, we didn't detect any avian influenza in penguin species whilst we were there, but we did detect an unusual mortality event at one of the locations. Samples there so far have tested negative, but we're sort of doing more tests at the moment.
Sounds like you suspect that there's something. Could the virus be so lethal to penguins that it's killing them even though it's not detectable? Or, what are your thoughts?
We're not sure at this stage what it is. It is unusual just because of the number of adults that we found that were in pretty good body condition. But it could be a range of things. So it could be other infectious diseases or other sort of extreme weather events and so forth. So we still have some more tests to do to rule out avian influenza and also other viruses as well.
I saw a picture that you shared of the lab that Antonio had set up on board the ship. It looked like this was a pretty improvised trip, but it's really important for you to take the equipment to be able to test for the genetic signature of the flu.
Yes, it was definitely a very impressive lab that they brought with them and set up pretty much, right in the hull of the ship. The team on the boat were fantastic and put together the space for them to have the equipment, and it enabled us to have results within a couple of days, which was really important.
I think you put this together pretty quickly, this mission. It was a very urgent thing for you to do, was it?
Yes, we had a very unique opportunity sort of at the last minute to take this trip. And so, within a couple of weeks, we brought the team together and headed down to the Antarctic. So, it was a pretty impressive job.
Where exactly did the ship go? This is only a snapshot, I guess, of the whole continent. But, how far did you go? How many colonies were you able to visit? Give me a sense of, I suppose, the picture that you're seeing at the moment in that part of Antarctica.
We went down to the tip of the Antarctic Peninsula and travelled down south, what we call the Trinity Peninsula on the western side, and then headed over to the Danger Islands. So it was all up 22 days that we were travelling around the region, and we visited 10 different wildlife colonies during that time.
And some of them were fine and others you had these mass mortality events? Or, did you find samples in lots of places?
So 4 different locations is where we were able to confirm avian influenza. And we had, of course, the unusual mortality at Heroina. But then other locations we went to, everything looked normal. There were still lots of healthy animals around. So there's definitely locations that it hasn't arrived yet, but it has sort of arrived later in the season once a lot of the animals have actually left for the season.
I mean, it does sound like you started near, as it were, at the South American end of the continent. And this is, I think, the route where the virus would get into Antarctica. You can't say at the moment how far it might have spread around the full vastness of it.
Yes, so at the moment, we know it's definitely gotten to the Antarctic Peninsula. So we've had a number of confirmed cases now. So the first one that Antonio did at Primavera Station, but then down further south into James Ross Island has also been confirmed. And now our sites, which are around that James Ross area, so they're definitely areas we've confirmed. So it's definitely on the peninsula. At this stage, it doesn't appear to be further around Antarctica yet. But unfortunately, next season's a different story.
Well, that's what I was going to ask. Presumably over the winter, there's little you can even do to try and watch. But next year, when the next breeding season, presumably migration patterns start kicking in, that's when you get really worried again.
Yes, definitely. We still don't know a lot about the virus, how it may survive in the environment down in Antarctica, or what species might sort of keep it circulating during the winter period. So we are definitely concerned for next season, especially now that it has definitely arrived.
Do you have plans to go back and do a more thorough search?
Yes, definitely. So next season we hope to get back down south and start doing some ongoing surveillance of populations throughout the season.
As an ornithologist, is this something all you can do is watch and observe? I know you've been very worried about the virus getting in there and giving advice on how researchers such as yourself can avoid accidentally bringing it in yourselves. But now that it's there, is there anything you can do apart from watching?
There's not much we can do to stop the virus once it arrives. There's unfortunately a lot of the seabird species and a lot of the animals do move around and mix a lot. So once it's down there, it's very hard to stop it spreading. There's definitely more we can do sort of back outside of the Antarctic region, especially with our farming and our poultry areas where this virus originates from. So trying to prevent further outbreaks and further spread. But in terms of, down south, a lot is conducting more research and learning more about the virus, which species are susceptible and how it actually moves. There's still so much we don't know about it.
Since you've been back, I don't know if you've been able to catch up with some of the other stories. Last week we were talking about the fact that the virus had been getting into cattle in the United States, and that seems to be spreading and spreading. There are now several states and several herds where it's got to, which seems extraordinary. I mean, this virus just seems to keep surprising.
It's definitely one that's coming up with new surprises every time you look at the media. There's something new, a new species or things that it's infecting. So, yeah, there's still a lot that we need to understand about it.
Megan, thank you very much for updating us. No doubt you won't be surprised. We'll keep following this as it spreads. That's Megan Dewar, who's based at the Federation University of Australia.
Professor Higgs, I now ask you to step forward to receive your Nobel Prize from the hands of His Majesty the King.
60 years after he came up with his enduring idea of the Higgs field and the better-known related Higgs particle, and 10 and a bit years after getting the Nobel Prize for it, Peter Higgs has died. If you've heard the tributes already, the theme has been about his aversion to the publicity that accompanied his celebrity, underlined perhaps by this comment to Jim Al-Khalili on The Life Scientific.
Well, the big event was really the announcement at CERN in July 2012. That was really the end of a long wait for me. As you say, it's been most of 50 years, and the experience of the seminar at Geneva was quite amazing.
Was it an emotional time?
Yes. So I'm not sure to what extent that was a result of the actual announcement. I rather think it was partly the result of the way the audience responded to it, which was to get up and cheer and applaud, and it was a really amazing occasion.
I never met Peter Higgs. As I say, he never sought publicity, and many others were very eager to extol his work to me. But physicist and historian Frank Close did meet him many times while working on his biography, as he told us in 2022.
Well, the title elusive refers both to the nature of the boson, which took 48 years from the original idea before it was confirmed in experiment, and also the nature of its creator, if that's the right phrase, in Peter Higgs, in that the Higgs boson is the one piece of work that he did for which he is known, and it is almost the only significant work that he did in the whole of his career. And that although he had the original idea back in 1964 and developed it in 1966 to give the sort of signature of what the boson would look like, he did nothing more with it in the subsequent 50 years. It was others that built upon this and developed it and brought it to the climax that happened just 10 years ago.
It feels almost like that he's some kind of shooting star that passed through the atmosphere of some quite important theoretical discussions in the early 60s. He sort of arrived when those discussions had started, and then he sort of flew off, left this bright trail behind that others picked up on, but he sort of vanished from those conversations.
I think that is a quite apt summary, in a way, of the whole saga, that Peter Higgs had this great idea in 1964. In fact, as he described it to one of his students who came back from summer holiday to find a note on his desk from Peter saying, "This summer I've had the only good idea I've ever had." And it was indeed a singular idea that happened to touch the very heart of how we now know the universe works. If we were able to look back, in 100 years on the course of 20th century science and the beginnings of the 21st century science, I think without a doubt the discovery of the Higgs boson will be seen as one of the seminal moments in human culture that ranks with the discovery of the structure of the atom itself.
This is very complex maths that we're talking about here, that it's very difficult to summarise in a programme like Science in Action. But does this sort of speak to some kind of penetrating insight that Peter Higgs was able to bring to these conversations about the nature of particle physics forces?
He had been trying to understand some technical features of quantum field theory for some time. The words are gauge invariance, to give it a name. And this was something that was not very well understood, and Peter was really trying to understand it in depth. And in the course of that, he, by chance, if you like, he'd been asking himself the right set of questions that when something when a paper arrived with a question in it, Peter almost uniquely happened at that moment to be in the right place to say, aha, I have the answer. Now, some people have said, well, no, was that luck? And I draw the famous analogy with Gary Player, who apparently having holed an almost impossible shot on the golf course, somebody said, Gary, that was lucky. And he said, yes, and the more I practice, the luckier I become. I mean, in Higgs's case, he had done the work for several years trying to understand things so that he was in the right place when chance arrived, and he was able to use that fortune to solve the problem.
Frank, if I can bring it back to the book, so we are 10 years on now from the discovery of the Higgs. There were a load of books that came out at the time. What was it that made you want to tell this story again in your own words?
I've always felt that, if you like, the challenge and the art in trying to popularise science is to be able to imagine what is it that the public perceive that I'm doing? How can I describe this in a way that will be meaningful to them so that they feel also it is worthwhile doing and then being involved with and excited by? And so on this occasion, it was doing this in the guise of Peter Higgs. And it's been a very interesting one. I talked with him about how he saw things before the boson was discovered and immediately after the boson was discovered, when suddenly he, who has always been a very modest and retiring person, was thrust into the limelight. In a very strange way, I sort of ended up feeling sort of quite, not exactly sorry for him. You can't be sorry when somebody's created such a wonderful edifice which will go down in history with their name on it and won the Nobel Prize. But he did say to me, I asked him, how did he feel after all of this had finally been done? And he said, it ruined my life. And that quite shocked me. And I asked him again the next time we spoke to be sure that I hadn't misunderstood anything. He explained, I wrote it down, and it's in the book, and I will read it carefully so it doesn't get misinterpreted. My relatively peaceful existence was ending. I don't enjoy this sort of publicity. My style is to work in isolation and occasionally have a bright idea.
Frank Close, whose account of Peter Higgs' life and work is far and away the best I've read. As for the science, I put together a series of Discovery editions in 2022 on where the research has got to and where it's going. We've put a link to that on the Science in Action webpage at bbcworldservice.com. And CERN's plans for a future circular collider we talked about recently, the chief idea of that is that it would be a Higgs factory, because the particle is a kind of portal to all other kinds of physics, if you can make enough of them. It'd be running long after the centenary of Peter Higgs' original paper, if they build it. And from physics celebrity to physics disgraced, concerning superconductivity, a real phenomenon that, for what it's worth, helped inspire Peter Higgs' theorising. Science headlines have been littered in recent years that this unusual state of zero electrical resistance had been reached in a range of hydrogen-based materials without the usual drastic freezing needed with known superconductors. The claims had the potential to be technologically transformative, if true. But as we heard from science writer Dan Garisto last November, those claims had fallen apart. And according to an internal university report he's now obtained, as we suspected but couldn't say, they were based on data, quote, falsification and fabrication. Dan, thanks for joining us again. I have to say, this really does look like a disgraceful episode.
You're right. As you said, it looked like that in October when I joined you. But the latest investigation report is really important because it puts this official stamp. This is really the first official public conclusion that Diaz has committed manipulation, specifically fabrication, falsification, and plagiarism. And having that be public, I think, or I hope, and I think a lot of other scientists hope, will help close this chapter on this misconduct and allow people to get back to researching superconductivity in other ways.
And as you say, so this is Ranga Diaz is the name of the scientist. He's been investigated at Rochester University, but he's also been at other universities. So how much of the data have they been able to get hold of to sort of narrow down the idea, to do that sort of paper trail of what the manipulation was?
Right. So Diaz got his PhD at Washington State University, and the investigators looked at his thesis where concerns had previously been raised about plagiarism, and they concluded that many, if not all of those concerns were correct. And they also found another instance, more recent, where he plagiarized in his 2020 NSF proposal from colleagues at Rochester. Then Diaz was at Harvard, where he worked on metallic hydrogen. The investigators didn't look deeply into this, but they did find that there were concerns about the reliability of the data there, but they did not conclude that there was misconduct, and I think it was sort of outside the scope of their investigation. And then in 2017, Diaz came to Rochester, and the investigators looked at 4 papers. One had to do with Diaz essentially copying data from his thesis and passing it off as another material. 2 of them had to do with this material CSH, which is under very, very, very high pressure. And CSH stands for carbon, sulfur, hydrogen. And Diaz was claiming that this was the first room temperature superconductor. Then the final one, and this is really where the data comes in, because when he was asked to provide raw data that is obtained directly from measuring instruments, he could not do that for so many of his claims. And if he had been able to do that, he probably would have been able to dispel many of those allegations of misconduct. But with this biggest blockbuster result, this lutetium hydride paper, Diaz claimed that he had made a room temperature superconductor at moderate pressures. And he was touting this as a route to something that would work in your living room. And the investigators were able to secure the real raw data from the hard drives and from the machines in his lab. When they looked at it, they found what they described in many cases as profuse manipulations. Some of the most striking examples were where he literally inverted the signal. So he made it show the opposite of what it was actually doing.
But it looked good. I mean, the phrase is that extraordinary claims need extraordinary evidence.
Yes.
But in a way, I do wonder, he's had a pretty celebrated career. Extraordinary personality, has that been a substitute? Have you ever met or talked to him? Is he sort of very persuasive?
I spoke with Ranga once before this. I was doing an obituary for Neil Ashcroft, who is sort of a father of the field of hydride superconductivity starting in the 1960s.
Character.
Yes. Anyways, when I spoke with Ranga about this, he was very gracious and very charismatic. And I think that there are a lot of people in the community who felt like he was this talented, rising young star who could transform the field of superconductivity and really bring it into the 21st century. It was hard for a lot of people to see not only that was not true, but that he has hurt a lot of people. First and foremost, his students. But additionally, as you said, these actions have discredited a field. And that's a shame.
It's the young researchers who are sucked into his orbit who I particularly worry about. Have you spoken to them?
I've spoken with a number of them. I had these now former students telling me, I'm worried about how I'm going to graduate with potentially fabricated data in my thesis. I'm worried about what Ranga will do if he finds out that I'm talking to a journalist or if he finds out that I'm helping the investigation. And many of those fears were confirmed last September when the students decided enough was enough and they were going to send a letter to Nature requesting a retraction of this lutetium hydride paper because Diaz sent them cease and desist letters. A legal threat to say, do not speak up about this. I think that was really, in some ways, a breaking point. And if the students had been cowed, who knows? I mean, I think eventually most of these things would have come to light, but I think it was very important for them and very important for the scientific community that they proceeded forward and they went to great lengths to uncover the manipulation that their advisor had made them party to.
I think it's so important. Congratulations to them. Also, thanks, Dan, for talking to us. Science depends so much on trust, and it does work like that most of the time, but there will be more scandals like this, no doubt. Meanwhile, we've put a link to Dan Garisto's latest reporting on our Science in Action webpage. Lastly, a single-celled seaweed, an alga with unique powers. You surely know that plants' special power is to capture CO2 from the atmosphere and break it up to get the carbon they need for their sugars, fats, proteins, all their biomolecules. And when we eat, we benefit from that superpower. But the atmosphere is 80% nitrogen, which is another essential element, yet plants have no ability to capture that. Instead, we depend on humble soil bacteria to do it, and hope that some of it reaches us in the end. Hence the interest in Rudosphera bigeloii, if I pronounce that right, a single-celled alga that floats in the sea and has co-opted a bacterium to get that all-important nitrogen for it, as marine biologist Tyler Cole discovered.
In the world's oceans, nitrogen can be limiting the growth of the organisms at the bottom of the food chain, the phytoplankton, in large regions of the world's oceans. And therefore, organisms that can perform nitrogen fixation and convert atmospheric nitrogen gas into usable forms of nitrogen, providing the fertilizer for plankton to grow, they can be really important in the ocean, and that's why our lab has spent decades really studying these organisms.
And a lot of the time, I guess, what's happening is they're just capturing the nitrogen from the air, incorporating their own bodies. Their body, they die, they decompose, and other things can then pick up the detritus. That's the normal route, I guess, quite a lot of time.
That's one mechanism. They absorb nitrogen, that gas that's dissolved in seawater. They perform this complicated process of splitting it apart. It's very energy intensive. And then that nitrogen can be shared with other organisms in the food web. It can even, in some cases, diffuse out of their cells and be shared, with their co-occurring microbes.
Now, the other aspect of your work is you're talking here about an organelle, and I'm thinking here of, the classic for me would be chloroplasts, which are captured bacteria inside plant cells that capture light and capture carbon dioxide and start making things like that. Or mitochondria, they're the sort of the energy factories of our cells.
Yes, the chloroplasts and the mitochondria are the classic examples of the result of this process that's called primary endosymbiosis, or as you mentioned, the capture of bacteria into a larger cell that then become a part of that larger cell. Those two organelles evolved in the distant evolutionary past, over 1 billion years ago, and the details of how they evolved are largely lost. We don't know how that process occurs. With this new organelle, the nitroplast, we have a new perspective on that process, and hopefully it'll provide insights into the evolution of these other very important organelles, which really, when the mitochondria evolved, it revolutionized life on Earth. It gave us all complex life, which it was then descended from it. Likewise with the chloroplast, when that organelle evolved, it gave us all plants and many algal lineages. So very important events in the evolution of life on the planet.
When you talk about a nitroplast, then you are talking about very much the same thing. This is some kind of primitive bacterium, is it, in the sea that's been absorbed and lives and works with inside these little algae cells.
That's right. It's very much the same thing. It's a bacterial cell that has been incorporated into a more complex cell, and it's lost the ability to survive on its own, and it's dependent on genes that are encoding proteins in the host's genome. So it no longer has a functioning genome of its own. It has a genome which is split between itself and the cell that it lives with.
I mean, the tone of your description in the papers, you're quite surprised to see this.
Yeah. It was a striking discovery to see. We've known about these structures within these algal cells for a long time. We just didn't have the ability to do the type of science which would reveal that they are indeed organelles rather than symbionts.
I mean, I always have this feeling that it's like a a merger between two companies, and that in this case, the organelle has outsourced some of its sort of office functions to the main cell, but it just keeps doing the bit that it's really good at.
That's a good analogy. The organelle has given up the ability to perform basic functions that most cells do, and certainly related species of bacteria would do if they were living on its own. In exchange, it receives proteins and metabolites from the algal cell, which allow it to perform nitrogen fixation very efficiently.
To me, one of the extraordinary mysteries of nitrogen fixation, getting the nitrogen from the atmosphere into organisms, get it into the food web, is that essentially, I think it was sort of one enzyme that was invented about 3 billion years ago and has just sort of mutated since then. It's an incredibly hard piece of chemistry to do, and it's been incredibly hard for a lot of organisms to capture. I'm wondering what lessons there are from the observations you've been doing.
Yeah, well, I think you're absolutely right. It is a energetically demanding process. It requires a lot of energy from the cell to drive nitrogen fixation. What the nitroplast represents is a new way of driving that chemical reaction. Nitrogen fixation always has this high energy demand, and different organisms can meet that demand in different ways. This nitroplast uses a novel coupling of it to light energy harvested by an algal cell.
I mean, it's a fantastic union of of function, as it were. To me, it seems, if only we could do this. If you could put these in your lungs or something like that, we wouldn't have to eat meat anymore, for example. We could make all our own amino acids and all our own proteins with no trouble.
Yeah, it is. It does impart a novel capability to these cells. With this, these algal cells are now capable of both carbon and nitrogen fixation, so they can kind of make all the nutrients they need.
Tyler Cole of the University of California, Santa Cruz, whose analysis just appeared in Science. And I was only joking about getting nitrogen through our lungs, but on the other hand, we'll be dreaming more science in action here on the BBC World Service same time next week. Until then, from me, Ronan Pease, and producer Jonathan Blackwell, thanks for listening.
Hi, I'm former Wales international footballer Robert Earnshaw.